thanks for coming. I really appreciate actually being in this plasticity section because, as you'll see, I didn't start out this work looking at plasticity, and I really have just started thinking about it, and I'd love to think about it more. So if any of you guys are experts in plasticity and you want to talk about this, particularly what I'm going to talk about now with me after this session, I'll buy you a beer. So come see me. Okay, so the question I'm more used to asking is this question, which is, what is the genetic architecture? How many genes, what do they do, what effect do they have of ecological speciation? And you can see patterns across systems of what we think of as putative or incipient ecological speciation systems. So there's a lot of theory out there about this question, and some of it suggests that the ideal architecture for ecological speciation, specifically with gene flow, with migration, is a magic trait. And when we use the phrase magic trait, we're often talking about essentially a magic locus, sort of a single locus trait. But it's a trait that is both under divergent selection and simultaneously contributes to reproductive isolation. So you might think of, for example, flower color. If there's pollinator preferences for different flower colors, having different flower color might change who pollinates you, which changes your opportunities to mate with other individuals of a different flower color. But for me, the system's a little bit different. So this is Helianthus argyphilus, the silverleaf sunflower. This is a system I work on for my PhD with Lauren Reisberg. And I first grew this species and noticed this really striking thing. So if you're not a plant biologist, I put in an example. So these are the same species, and they're from a relatively small range for the genus. But some plants, when I grew them in the common garden, just a few populations, they grew until about mid-June. They started flowering. They're annuals, so they've stopped growing. And they're about 1 meter tall, and they continue flowering, put out more flowers throughout the season. And then other populations, other plants just kept on growing. And this photo was taken in early October, when you can just see the first flower head forming on this plant, which is about 3 meters tall at that point. And that's pretty significant. So like the dogs, they're the same species. You can cross them experimentally, but there's some amount of reproductive isolation here, right? Like, depending on how you're trying to do it, this guy and this guy might have a hard time. Okay, so these guys are from Texas, just in case you don't know where we are. We're here. I made a big star because we're in Texas. So they almost get up here, but they're pretty much a little bit farther south of us. So they're restricted to the sandy soils, essentially along the Gulf Coast, down to Mexico, but we don't know much about this part of the range. And I'm going to quickly talk about the range a little bit. So they're found on these thin barrier islands that run along the Gulf of the U.S. You can think of them as overgrown sandbars, pretty much just sand on top of water. They're also found along the mainland coast in beachy areas, and they extend inland. They're on sort of riverbeds, river bottoms, and sandy plains. Okay, so I'm going to summarize a lot of work very quickly, but essentially, I went out, I collected a bunch of plants from across the range in all of these places I talked about, and I grew them in the common garden. When I grew them in the common garden, I measured a number of traits, 20 traits, and they essentially sum up to a single trait, which I am calling a life history syndrome. So there's one cluster here along this first axis, and one cluster here along this first axis, which explains about half of the variation among individuals. And these clusters are a lot of traits that co-vary. So on this end, you have things that are early flowering, that are relatively short, about 1 meter. They make a few branches, they have really large flower heads. And on this side, things are tall, late flowering, many branches, small flower heads. And there's an important thing to note here. I've colored the individual data by the region of origin for that individual. And then I put the regional means here in these little triangles. So you might notice something, which is that pretty much everyone is over here, which is this late flowering cluster. That's the plants from the North Inland, from the mainland coast, so this is near us, and plants from South Inland down by Brownsville. And then about half of the plants from this island region, the barrier island region, are in this other cluster. So there's some regional variation. Yeah, that's just showing you, that's this cluster, that's that cluster. But genetic variation, genetic structure is not sufficient to explain this variation. So again, The difference is this region versus these two regions and this region. So we see structure in this species, but it's not aligning with the phenotypic variation that we see, in the sense that these two regions are phenotypically differentiated, where early flowering plants and late flowering plants are both found here. These guys are almost exclusively late flowering in this particular analysis. But they're not genetically differentiable in these data. And if you're more curious about this set of data about this life history syndrome, this just came out early view online this week, so you can go read about it. Kind of exciting. I wanted to note two more things, actually. One is that here, it's important to note that tall things are late flowering because they keep growing until they flower in this environment, and early things are short. And that this environment is not the native range of the species, but it was a pretty cush environment for these plants. Lots of water. I made sure to baby them in the early life history stages. So it's a low stress environment. Very few plants died. Pretty much everyone survived to flower. And tall things are short. Tall things are late. Short things are early. So the typical next question in my field is to ask this. What's the genetic architecture of this life history variation? So potentially it's contributing to reproductive isolation. Maybe you want to know what the genetic basis of it is. So I did this using two replicate F2 mapping populations that were across two separate regions. Again, I evaluated them in a low stress environment, and this is the key thing. This is the same sort of trait space to show you here, is that I recapitulated in this F2 exactly the same phenotypic distribution, which is essentially, again, these two modes. So in this case, these guys are early, these guys are late, but there's two modes on this first axis, which explains most variation, and along which most of the traits covary. And when I do linkage mapping on these traits, this is why. So I see incredibly strong plasticity. So here I'm just showing the scan1 log traces for support for QTL, any locus across different chromosomes. The little hash marks are markers. And the colors here are traits, and then these are the two populations. I'm only showing chromosomes for which, or linkage groups for which we had significant QTL, different numbers. But the key thing is this, look, huge support for all 6 traits in both populations, and for many of the traits, the only QTL we identified under multiple QTL mapping modeling was here at the end of linkage group 6. Okay. Very quickly, we have a really good candidate for something that could be a single gene that's responsible for this variation. This region is syntenic with a region on the annuus, the sister species chromosome, which is the site of several known major flowering plant QTL, and contains 3 paralogs of this important flowering signal integrator, which is a really good place to start looking for candidates. So I'm going to skip over this for now. What I want to focus on is that, given what we know so far, this life history locus is a great candidate for a magic trait, right? It's one locus, it can't be broken apart potentially, or they're tightly linked things that are difficult to break apart by recombination. And it's a site that potentially is under divergent selection, but definitely creates reproductive isolation. If you're flowering 5 months apart, it's difficult to mate. Okay, so the next sort of logical thing in my chugging along in the sort of standard way in the system was to ask if it was under divergent selection. So I skipped a slide? No? Yeah. Just put them out of order just to make it fun. Okay, so we went out down to South Texas, around near Corpus Christi, and we planted, we tagged 150 plants, wild plants, we didn't plant any of at 7 sites that span the range of this species. So we have 3 sites on the Barrier Islands, 3 on the mainland coast, and then 2 on the mainland coast and 2 that were inland. So at each of these sites, for each individual, we recorded when they flowered, if they survived, when they died, their height at flowering, and then fecundity as the number of flower heads they produce at the end of their lifespans, as a component of fitness, or proxy for fitness in the system. Okay, so this is what we see. And what I'm showing here, I'm going to show you this a few times, so take a second here to talk about it. But on the x-axis here is flowering time. On this side, you're early. On that side, you're late. On the y-axis here is height at flowering, so short and tall. And drawn on here are these contour lines for the population mean fitness in this trait space, this bimodal trait space. I find these contour plots sort of difficult to see, and the font is kind of low. So I made a cartoon just to show you the evidence here. So what I've drawn is sort of where you would go if you were a population mean moving around in space driven by selection. There's no constraints, right? So everywhere that we did this, it was better to be taller. But in some places, it didn't seem to matter when you flowered. It's more or less flat across flowering space. In some places, there was selection on the barrier islands where we see early flowering plants, or selection for earlier flowering. And on the nearby coast, where you see mainly late flowering plants, there is selection for later flowering, maybe moderate late flowering. And an important thing to note is that these are each scaled to the population mean and variance. So if I plot them together, this is a cartoon, the space that these selection landscapes, these citrus landscapes are exploring is quite different. But the key thing to take away is that there is divergence in selection on these traits space across the three regions, two of which phenotypically are similar in the common garden, and one of which about half the individuals look different and are down here versus up here. But they're, okay, so given this is a pretty good magic trait, potentially. In theory, predicts that strong divergent selection on this trait should lead to speciation, or at least increased reproductive isolation, the evolution of stronger barriers. Okay. So there's a problem, though. If you look at the actual phenotypic space occupied by individuals in this experiment in 2011, we're missing two things. One is that we haven't recaptured a bimodal distribution. Maybe a hint of that here, but there's really just one pulse. And what's more interesting to me is that we've lost this positive correlation between flowering time and height at flowering. In the sense that here it's about neutral, here there's a negative relationship, and again, here there's a negative relationship. So we lost the magic, right? And I had a hypothesis about this because I did this experiment in 2011, which was both independently the hottest year in record for Texas